4. All right, before I share my screen, let me talk a little bit about the grading I've done. The Plagy quiz is graded. That's done automatically. The extra credit study sheet's graded. I think I graded those last week. Worksheets, lectures 1, 2, and 3 are graded. When you're doing the lecture worksheet, make sure you look up the answer. If you do not, absolutely, you're not sure of the answer. And I graded lab 00 and lab 01 for the second time. And I've graded lab 2 and 3 and 4 for the first time. Any question about any of that? I do need to grade quiz 1A and quiz 1B. You guys have taken, it's graded automatically. I'll go in and take a look at it. All right. Any questions on anything? If not, let me get out of the grade book.
Do we have the answers for the quizzes yet?
I think so. I think you get them like 2 days afterwards. And I think you took the quiz. Is it on Wednesday you take quizzes? I'm getting a little confused here. Let me see. You took the quiz on Wednesday, so the answer should be ready midnight on Friday. So
Okay, I'll take a look. Thank you.
quiz 00, the answer should be up. Quiz 01, the answer should be up for quiz 01 also. I mean, quiz 1. Now for the multiple choice quizzes, you have to get in it with the lockdown browser. So respond as lockdown browser, get in the quizzes with that, and you should be able to get the answers that way. For the short answer quizzes, let me see if I can show that. I've got a link to the short answer quizzes. Let me show this now. Share my screen. So for the short answer quizzes, come to this link here, quiz 1 answers, quiz 1A answers, and they should be showing from January 21st to January 28th. I guess I can make that a little longer. Let's go ahead and make that February 4th. All right. So let me come back to the syllabus and say that today is January 24th. We are supposed to be covering chapter 4. I will look and see. Did we finish chapter 3? Anyone know? Nobody knows? Let me take a look at that. Make sure we finish chapter 3. Staining.
The last notes I took were about gram-positive and gram-negative bacteria and how capsule stains are useful for unknowns project.
Yeah, I talked about the special stains, the capsule stains. So we finished chapter 3. But thank you. I was trying to remember, and I do remember discussing special stains. So We're on chapter 4 today, which is good. That means we're on schedule. Chapter 4, functional anatomy of prokaryotic and eukaryotic cells. I will talk about the anatomy of prokaryotic cells and eukaryotic cells. I'm just going to review it. If we don't have time, I won't even cover eukaryotic cells because you should have had that in Biology 160. And so that should be a review for you. But the major goals of this lesson are, of course, to know the terms, what's a coccus, a bacillus, a spirilla, a strepto, like streptococcus, staphylococcus, diplococcus, a tetrad, Sarcenia, what's a vibrio, spirochete, hopefully you'll know all these already, glycocalyx, I think we talked about that, flagella, axial filaments, that's one I think we have to talk about, Fimbriae, and what's pili? Peptidoglycan, we'll talk about that in more greater detail. Endotoxins, selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid. What are inclusions? What are endospores? We've talked about this already, but we're going to talk about it at least 3 times this year, and we'll talk about it today. And then lastly, we'll talk about the endosymbiotic theory. This is actually in eukaryotes, but that might not have been a topic you have discussed, and we'll talk about that today. And then not today necessarily, we'll probably talk about that on Thursday. We'll also talk about how gram-positive and gram-negative cell walls differ and be able to describe the importance of taxis in bacteria. Any question about what we're doing? As you recall, to know all of the objectives, take a look at the study guide to chapter 4, and the start of that will have all of the objectives for chapter 4. So on our first slide, we're going to compare prokaryotes and eukaryotes. Pro is the Greek word for prokaryotes, is the Greek word for pre-nucleus. Strictly speaking, it's pre-kernel, but it means pre-nucleus. Eukaryotes, Greek for true nucleus or true kernel. And you don't need to know kernel, so we'll just say nucleus. They differ in their chromosomes. Prokaryotes typically have one circular chromosome, and that chromosome is not in the nucleus. There is no nucleus in prokaryotes. Eukaryotes have typically many chromosomes. They are linear, not circular, and they're inside the nucleus. Prokaryotes have their DNA associated, they lack the DNA associated with histones, and eukaryotes have their DNA associated with histones. Histones are a protein that are a major part of the chromosomes. There are other proteins in the chromosomes, but histones is probably the number 1 chromosome protein in the chromosome. And as you don't know, chromosomes are both DNA and protein. Even prokaryote chromosomes are DNA and protein, but there's more DNA in eukaryotes, and not more DNA, actually there is, but there's more protein in eukaryote chromosomes, and that's largely due to the histones. There are no organelles in prokaryotes. There are organelles in eukaryotes. By organelle, I mean a membrane-bound organelle, and that's kind of a mouthful, so I usually just say organelle. And there are non-membrane bound organelles, and those are terms that your textbook uses. I tend to just call them organelles for membrane organelle, and then a cell structure for a non-membrane bound organelle. Any question about that? Okay, in bacteria only, you can find peptidoglycan in their cell walls. There's no peptidoglycan in the cell wall of any eukaryotes, including animal cells that do not have a cell wall. Prokaryotes mainly divide by binary fission, where the cell divides into two, and eukaryotes, they divide by mitosis. Any questions? So we're going to talk about prokaryotes, their cell shape and cellular growth arrangement, their arrangement. We'll then move on to their cell structures, talk about the cell structures, then move on to the movement of materials across membranes. And then if we have time, we'll start talking about eukaryotes, their cell structures. If we don't have time, I won't discuss it. And we will talk about the endosymbiotic theory. Okay, when we're talking about prokaryote cell sizes and shapes, you should realize that their average cell size is actually around 1 to 2 micrometers, but you can get very big prokaryotes that get bigger, like 10 micrometers. And the very smallest are 0.4 micrometers. The basic prokaryote shapes, they have 3 basic shapes. There are coccus cells, and they're round, like a ball. There are bacillus, and these are rod-shaped. And there are spirals, shaped like an S. The 3 basic shapes of prokaryote cells. Now, there are prokaryotes that have different shapes, but most of them are some derivative of these 3 main shapes. Talking about the spirals, and actually a vibrio, you could call it a half of a spiral, but you could also call it just a bent rod. It doesn't really matter. A vibrio is a curved rod, or you can say a half of a spiral. There is a difference between a spiral and a spirochete. A spirillum is a rigid structure. It does have a corkscrew shape or an S shape. And the spirochete is similar. It's just that it's got more, I don't know, bends or spirals in it. But it's a little different in that it is a flexible structure. A spirochete is a spiral, but it's a special spiral. There are some prokaryotes which are unusual, like the star-shaped Stella bacteria. Actually look like that. That's a real picture. And then the square or rectangular Haloarcula, that's an archaea bacteria, which there's an actual picture of one. Most bacteria species are monomorphic, meaning they have a single shape, star shape, rectangular, rod, but there are some which are pleomorphic, which they have many shapes. Here's a pleomorphic bacteria. Most of the pleomorphic bacteria are bacteria that lack a cell wall. Most bacteria do have a cell wall, but there's one family that lacks a cell wall, and we'll talk a little bit more about that. And all the members of that family are pleomorphic, meaning they take on many different shapes. When we're talking about prokaryotic cells, they have a cellular arrangement, sometimes called a growth arrangement, or even altogether a cellular growth arrangement. This is just how the cells stick together when they are growing. You can have singlets. So with cocci, you can have a singlet cocci, and there's a singlet there, and there's another singlet. I think that's a singlet. You can have diplococci, that's 2 cocci stuck together, and there would be a diplococci. I suppose that's a diplococci, and I'm not sure, those could be a diplococci there too. There are streptococci, and that's just when the cells are stuck together more than two, and they're in a row called the streptococci. There are cocci that take the tetrad shape. It's 4 cells together. And this is kind of hard to see. Let me see if I can blow that up. Still hard to see. Now, if you look there's too many of them, so it's hard to see that there's 4 right there. 1, 2, 3, 4. And I don't know what that bump is. Here's another 4 there. And then there's Sarxinia. Those are 8 cells stuck together, which you probably can't see 8 because there's too many cells to count 8, but Sarxinia there. And you'll note that for each of them, they have a different plane of division. The diplococci and the streptococci have one plane of division. And that's why the cells will line up in a streptococci. The tetrad has 2 lines of division, meaning they'll divide on this plane and that plane, forming the tetrad. And then the Sarxinia divide on 3 planes, giving rise to the Sarxinia. And then the staphylococci is the last cellular growth arrangement of cocci, and it actually means cocci in the shape of a cluster of grapes. And it has the same growth arrangement as the Sarxinia, meaning it can grow on any plane. You don't really need to know the plane of division, it's just showing you. The bacilli have many of the same cellular growth arrangements or the cellular arrangements as the cocci, but they do have fewer of them. Let's see if I can blow this up. There are singlets, so single bacilli, and there would be one there. There's diplobacilli, and that's 2 bacilli stuck together on their long end. There's streptobacilli, and that's a row of bacilli stuck together on their long end. If the bacilli are stuck together on their short end, we call them a palisade. And that's because, let me see if I can get this. That's because it reminds people of a palisade fence. I have a feeling I don't have a link to this. I should close down some of these things. Paint. Mark Twain actually was writing about a palisade fence. And what you have is you have a bunch of boards stacked this way, and this goes into the ground. And that's a palisade fence. If you remember reading Mark Twain, I don't remember if it was Tom Sawyer or Huckleberry Finn, probably Tom Sawyer was painting the fence. And he was talking about how great it was. And it was a palisade fence, if you don't remember. Let's remove that. Now, of course, this is the ground, this side, but any questions about that? Make sure that I'm recording. Yes, I am. There is a different shape of bacteria. I'm not sure why it's under a bacilli, but there are a few bacteria that are coccobacillus. And that is if you have a bacillus that's rounded on the edge and you make it shorter and shorter, it'll eventually become really cocci. But before it becomes a cocci, it'll be in between a cocci and a bacilli. And here, I'll try and blow that up, is viewing cacao bacillus, where they're a little short for a rod and a little long for a cockeye. You should note that the term bacillus has two meanings. There is a bacteria, which is a bacillus, a bacteria shape, which is a bacillus. And if it has a capital B, that means it's a family of bacteria, meaning a lot of species in the families. So that's a family if it has a capital B. Lowercase b is a shape. And then the same, don't confuse, where do we have it? Streptococci. That is a cellular growth arrangement. And if you see it, Streptococcus, that is a family of bacteria. Any question about that? And the Streptococcus family or the bacilli family will always be capitalized. The bacillus will be lowercase unless it's the start of the sentence. And Streptococci, of course, is a little different than Streptococcus, but it's generally lowercase unless it starts the sentence out. All right, any questions about cellular growth arrangement or cellular shape? If not, let's move on to taking a look at the structures to a typical prokaryotic cell. You're actually seeing that now. The red items are structures you find in all cells. All cells have three items. They have a plasma membrane. They have cytoplasm, and they have DNA. Now, all cells do have ribosomes, but please understand that the ribosomes of prokaryotic cells are a little different than the ribosomes of eukaryotic cells, but all cells also have ribosomes. Let's talk about some of the other things we're seeing. A pilus or a pili is a reproductive structure. It protrudes out of the cell, like the flagella is inside the cell, or at least inside, attached to the membrane, but it also mostly protrudes out of the cell. The cell has the flagella for locomotion. The cell wall is the last or the outermost part of a cell. So anything beyond the cell wall, is outside the cell. Anything that's inside the cell wall is inside the cell. And it's kind of funny that the cell wall is the division of what the cell is, because the cell membrane, which is inside the cell wall, is actually the gatekeeper for what comes into the cell and what leaves the cell. But it's actually the cell wall is considered a part of the cell. A capsule or a glycocalyx, it's written out as capsule here, is something that is outside the cell wall. It is secreted by the cell, but it is considered outside the cell. It is not considered a part of the cell. Any questions about that? We'll talk about plasmid. That's a piece of DNA separate from the chromosome. Where you find the DNA in a prokaryotic cell, we call it the nucleoid region. And it stains a little differently, as seen here, from the rest of the cytoplasm. But there's no distinction of where the DNA is and where the cytoplasm is, because the DNA is just floating around in the cytoplasm. And wherever the DNA goes, that is where the nucleoid region is. Any question about that? There's no boundary between the cytoplasm and the DNA. Let's see, what else do I need to talk about? I think I've talked about everything here. I haven't talked about an inclusion. An inclusion is something that the cell is storing, and it is stored at such high concentrations that what it is storing is it precipitates out of solution. So an inclusion is something that is not in solution in the liquids in the cytoplasm. And the ribosomes also are so big they do not go into solution. The DNA, for most of the time, is dissolved in solution. An exception is when the cells are going through mitosis or meiosis, and those are eukaryotic cells that do that. They will condense, the chromosomes will condense and come out of solution. And that's when we see the chromosomes. When the DNA is in solution and you were to take a picture of it, you would not be able to see the DNA under a light microscope because it's in solution. Any question about any of that? All right, let's talk a little bit about the glycocalyx. That is a structure that's secreted by some prokaryotes. It is outside the cell wall. It's usually sticky, and it usually is made up of polysaccharides, carbohydrates. It's largely water, but it does have carbohydrates in it. There are three types of glycocalyxes. There is the capsule, and that's a neatly organized structure. It helps prevent the bacteria from being phagocytized by a white blood cell. If you can see the glycocalyx under the light microscope, it is a capsule. There is also a slime layer, and it's an unorganized glycocalyx, loose structure, mostly water. I mean, even the capsule is mostly water, but the slime layer is like 95% or more water. So it is mostly water, and it doesn't show up with the light microscope. There's not enough carbohydrates and other molecules in it for it to show up. There's also the extracellular polysaccharide, which is found in biofilms, and we'll talk a little bit about biofilms later in the term. That's something that certain cells secrete to surround themselves with a film of material, and it does help the cell survive. I'll just word it that way. It does a lot of things, but that's one of the things it does. It helps the cell survive. So three different glycocalyxes. The capsule is the one you'll most need to know about because when you're working on your unknown project, the cells may have a capsule. You can see the capsule under the light microscope. The slime layer, we'll hardly ever talk about it. You can't see this under the light microscope. You have to see it under the electron microscope. And then the extracellular polysaccharide, we'll almost never talk about that because we're not really going to talk much about the biofilms. All right, let's move on to the structure of the flagella. It's a structure used for locomotion. If a bacteria or an archaea can move, and I'm talking about swim in a liquid media, they have a flagella because that is the only way that prokaryotes can swim or move. The flagella is mostly outside the cell wall. It does have a small portion that's inside the cell wall, and then even a small portion, a very small portion, which is inside the plasma membrane. but most of it is outside the cell wall. Most of it is composed of the protein flagellin, which makes up the flagella. The flagella whips, or like a whip, and when it beats, the cell moves in the opposite direction. So this will move this way, and then the cell will move forward. Any question about that? All right. This is the 4 basic arrangements that a bacterial flagella will need. You don't need to know this for the quizzes, but you will need to use this slide or something similar for one of the labs. And you may have already had that lab. Yeah, you did. That was the last lab you did. So there's a monotrichus, there's a flagella on one side, amphitrichus, the flagella on both sides, and then lophotrichus, multiple flagella on both sides, and then peritrichus, flagella all over the cell, on more than just the sides. When you're talking about the flagella arrangement, you have to use the highest arrangement you see, because when the cell first starts making flagella, it won't make it as peritrichus. It'll first put out 1 flagella, and then it'll put out probably 2 flagella. I don't know if it'll be 2 on one side here or 2 opposite sides. I just don't know about flagella. And then it'll become lophotrichus. And then lastly, it'll become peritrichus. Okay. So as I stated, if a bacteria or a archaea has a flagella, it can move. So it's a motile cell. And cells can rotate the flagella where they can run or tumble, meaning they can swim forward or they can actually tumble themselves. I don't know if you can see what I'm trying to do. Maybe I can do it with my glasses. So swimming would be moving this way, but tumbling is when the cell just tumbles. And it does that because of the flagella. Taxis is the movement toward or away from a stimula. There's 2 types of taxis. There's chemotaxis, movement towards or away from a chemical, and phototaxis, movement either towards or away from light. And this one is phototaxis, but notice how the light source is up here and the flagella is on this side. So the cells are moving away from the light. In this case, usually, and certainly for the photosynthetic bacteria, they'll move towards the light, not away. But this is not a photosynthetic bacteria. It must be a, what do you call that? Zooplankton that is not photosynthetic and it's moving away from the light. There is a special flagella that is called an endoflagella, and that's usually the term I use. It is also called an axial filament, and it is a flagella that is inside the cell wall of a spirochete. So the endoflagella are only found on spirochetes, and it is anchored on one side of the cell wall, inside the cell wall, and we'll just say it's anchored right there. I don't know if it's anchored here or there because it's not showing there. And let me go ahead and get that link going because that'll take a while. But when we look at the cell wall, let me go back to that picture. Oh, I just didn't let that run because it's under motion. Shut that down.
When the stakes are high at work, your tone matters.
Shut it off. When we look at this, let me go from this here. There's the cell wall and the endoflagella. And as you can see on this lower picture right here, that axial filament or endoflagella is inside the cell wall. The light pink is the cell wall, and it goes around, encompassing the axial filaments inside the cell wall. And you'll notice the endoflagella tends to wrap around the spirochete. All right, let's go to the video. Skip the ad. And here we see a spirochete moving. You notice how it just can change direction. In fact, that did a real change there. Let's go back a little bit. That it just beats the other. Whoops, that's not far enough. So going this way, it just changes direction. It can go that. Then it bent and it switched from going north and south to east and west. So telling you how flexible the spirochetes are and how they can go. There it is. It has a kind of a corkscrew type movement. And that movement is actually important for human pathogens that are spirochetes. It's believed that the corkscrew movement can actually burrow through the mucous membrane and then get into the patient and infect them. Any question about any of that? All right. Next, we have fimbriae. These are little hair-like structures which are on a bacteria, and they are part of the cell. They protrude out of the cell wall. They are straighter and thinner and shorter than a flagella. There's usually more fimbriae than flagella. They are not used for swimming. They are used for attaching to surfaces. Okay. So we're not going to talk about them in movement. Some bacteria actually can use fimbriae for moving, and you don't need to know this, but they will attach that to the surface here, and then they'll retract it. That'll pull the bacteria forward. And we're not going to discuss locomotion that way, and we're only going to talk about bacteria swimming. So they don't use the fimbriae for swimming. They use it for attaching to structures. The fimbriae can be a pathogenic structure if it aids a pathogen attaching to its host cell. Pili, also called sex pili, are longer than fimbriae. See if I can blow this slide up. In this picture, which is still kind of small, you can see that this is a sex pilus. That's singular, for pili is plural. And it's longer than the fimbriae, which you can see them, the hair-like structures here. Okay, the sex pilus is used to transfer DNA from one cell to another cell. And we'll talk a little bit more about this when we discuss chapter 8, genetics, but the sex pilus will allow DNA to move from this cell into that cell. Any question about any of that?
Would you mind repeating that just one more time? I don't know if my internet connection went out or you cut out, but I couldn't hear anything.
The sex pilus is used for transferring DNA from this cell to that cell. And at the moment, you don't need to know which cell the DNA moves from, but the point is it moves from one cell into the other cell. Okay. And we'll talk more about that. And it's usually a plasmid that moves across the sex pilus. All right, let's talk a little bit about the prokaryotic cell wall. Most prokaryotes do have a cell wall. Not all of them do, but most of them do. The cell wall of a prokaryotic cell is complex. It's a semi-rigid structure. It's responsible for giving the cell its shape. Like if a cell is a bacillus, it's because the cell wall is shaped in the shape of a bacillus. The cell wall is important because it protects the cell and prevents osmotic lysis. If the cell were to be put into an environment which has more water than the cell has, water will start moving into the cell. And if the cell doesn't have a cell wall, the cell membrane will just keep expanding as more and more water moves into the cell. Well, eventually, the cell membrane will pop. It'll be too big and it can't stretch any further, and water keeps moving in, and that'll pop the cell. And then the cytoplasm will leak out, and that will kill the cell. If the cell, on the other hand, has a cell wall, the cell membrane can expand only up to the cell wall. The cell wall will prevent the cell membrane from expanding any further. And if the cell membrane can't expand any further, no more water can move into the cell because there's no room to hold the water. So no more water can move into the cell. And that will prevent the cell membrane from bursting, which will prevent osmotic lysis of the cell. So the cell wall is for protection, and it prevents the cell from undergoing osmotic lysis. The cell wall is located outside the cell membrane. It can contribute to the pathogenicity of some species, like the mycobacteria have molecules in the cell wall which aid the mycobacteria in being a pathogen, assuming the mycobacterium is a human pathogen. And it depends on the species, but if it is, there could be molecules in the cell wall which can aid the cell to being pathogenic. And that's true for other molecules and other species. I just don't know off the top of my head one. Peptidoglycan is the main molecule in the cell wall of bacteria. Remember, peptidoglycan is only found in bacteria. It is not found in eukaryotes. And in most archaea, it's not found. You don't need to know the last part, but for our purposes, we're just saying peptidoglycan is found only in bacteria. Peptidoglycan is a polymer of a repeating disaccharide. And the two saccharides, which I guess are monosaccharides, are NAG and NAM. Specifically, let's see if I can blow this up so you can read it. NAG is N-acetylglucosamine, and NAM is N-acetylmuramic acid. And these names probably mean as much to you as NAG, so you might as well call them NAG and NAM, but you can call them by the long name, I don't care. What they are disaccharides linked together, one after the other. So NAM, NAG, NAM, NAG. And then they're repeated. There's a row of it here and a row there and a row there. And that's a plane. And underneath this plane, we have another plane. And underneath that plane, we have another plane. You getting the picture of peptidoglycan? Anyways, that's the saccharide part of peptidoglycan. The saccharides in their chains are linked together with a polypeptide. And the polypeptide is linking, come on, this chain to that chain and that chain through this polypeptide and then that polypeptide. And you notice this polypeptide links to that polypeptide. And I think this polypeptide is linked to this disaccharide chain, and there's the other disaccharide chain. And so these two are linked together through that peptide and that peptide and this peptide. And these two are linked together by this peptide. Any question about that? So we have polypeptides and disaccharides, and that actually gives peptidoglycan its name. Glycans are sugars, so NAG and NAMs are glycans, sugars, and it has a polypeptide in it, and that gives it its name peptide, so peptidoglycan. Any question about that? I just want to briefly mention that the peptidoglycan linked together by the polypeptides is actually the way that penicillin inhibits bacteria, leading to the death of the bacteria. Meaning that penicillin, when the cell wall is being put down, penicillin gets in the cell wall and blocks the formation of the polypeptides holding the disaccharide chains together. And so when the cell wall is being made, we have, instead of a peptide holding the disaccharides together, we have penicillin in there. And that creates a hole in the cell membrane. And what happens when you blow up a balloon in a box and the box has a hole in it? The balloon will start protruding out of the hole in the box. And as you keep blowing up the balloon, eventually the balloon will protrude out of the hole so much that the balloon will pop. And that's what happens with the cell membrane. Water will move into the cell, and the cell membrane will protrude out of the hole in the cell wall that penicillin made. And the cell membrane will start protruding so much that eventually it'll pop, and then that will kill the cell. And that's actually how penicillin kills bacteria. All right, I see what the problem is. I don't know if you guys can hear me. I have an unstable internet connection at the moment. Sorry about that. Let me see if I can shut some things down and see if that helps a little. Nope, don't want to do that. I probably don't need this any further, so I'm going to shut that down. And I don't really need that more. So let me shut that down. All right, how are you guys hearing me now? Can you guys hear me?
It's a little
Better.
Okay. All right, I'm not sure what's wrong with my internet connection, but it's unstable at the moment. Hopefully it doesn't die. If it does die, I will try and come back. And you guys don't need to do anything if I am disconnected. But if I'm disconnected, I will try and come back. All right, let's talk about the bacterial cell walls. The bacterial cell walls have two types, the gram-positive cell wall and the gram-negative cell wall. The gram-positive cell wall has a very thick cell wall shown in purple, and yellow is the cell membrane. And there we have a blow up of it. Let me see if I can blow this up a little. So there's the cell membrane and the cell walls outside of it. It's only showing you 3 layers of peptidoglycan. In the gram-positive cell, it's many layers of peptidoglycan. Okay? It's actually many layers of peptidoglycan. How many depends on the species, but by many, I'm talking about 100, maybe even 1,000 layers of peptidoglycan. It's many more than 3. All right. When we look at the gram-positive cell wall, we can say the main molecule is the peptidoglycan. The, I don't know, purple rods here. And then the short peptides linking the purple rods together. All right, those are the main molecules, which is peptidoglycan. But there are other molecules in it. There's also teichoic acid, which it's thought that it helps to hold the peptidoglycan together. And then there's lipoteichoic acid, which is the same as teichoic acid, except lipoteichoic acid has a lipid attached to it, and the lipid region of the lipoteichoic acid is bound in the cell membrane. All right, any question about that? That differs from the gram-negative. Oh, I need to say that the teichoic acid may help regulate the movement of cations through the cell wall. Cations are dissolvable in water, and anything that's dissolvable in water can actually go right through the cell wall. So the cell wall is not going to prevent things from entering or leaving the cell, at least anything that's dissolvable in water. But the cations might be able to use the teichoic acid for moving down the cell wall. The polysaccharides found in the cell wall can also give antigenic variation so that we can distinguish different strains from one another by their antigens. Like you probably heard of E. coli. What is it? H7O111. These are different antigens in E. coli, and there's different numbers. I don't remember the other numbers. I just remembered that one, but I think there's H5 and something like O121. Those are different antigens, and they're from polysaccharides in the cell wall. The gram-negative cell wall is different in that it has a thinner purple peptidoglycan layer, and it also differs. It does have the yellow cell membrane like we did with the gram-positive, but it has a lipid layer on the outside of the cell wall. And let's blow that up a little. Move over a little. There's the cell membrane. And in this picture, it's showing you the peptidoglycan as one layer. That is not correct. In the gram-negative cells, there are several layers of peptidoglycan, perhaps many, but it's many less than in the gram-positive layer. And by many, I'm talking about something like 20 to 30 layers. But it's many less than in the gram-positive cells. So it's more than one. So don't tell me that the gram-negative cells have a single layer of peptidoglycan. They have actually many, but this picture is only showing you one. Any question about that? All right. You'll notice that the gram-negative cell does have the layer of peptidoglycan, and outside that peptidoglycan, we have a bilayer of largely lipids. So it's similar in the sense to the cell membrane, which is a bilayer, but in the cell membrane, that lipid is phospholipids. And on the lower layer of the outer membrane of the cell wall, that is the case, that the main molecule is phospholipids. Okay. On the other layer, there are phospholipids, and they're shown in yellow, but there's another lipid, lipid A, which is the blue roundish structure, and attached to that lipid A is an O polysaccharide, which makes the hair, which means that the lipid A and the O polysaccharide are attached, and so we call that a lipopolysaccharide. and there are lipopolysaccharides in the outer membrane of the cell wall. The lipid A is the important part because lipid A is an endotoxin. We'll talk about toxins later in the term. I think we've already talked about toxins actually, but something that is a toxin is something that is toxic to us or poisonous to us. So we don't really want to be exposed to toxins. And if ever a person has an infection by a gram-negative bacteria, and if that cell has a cell wall, which most of them do, the patient will be exposed to an endotoxin because all gram-negative cells that have a cell wall have lipid A in their outer membrane of the cell wall. Any question about any of that? All right. There's one other thing we need to talk about. Well, maybe two other things. Let me remind you that the way that molecules move into the cell membrane is through a transport protein that spans the membrane. And then we have one specific protein to move one specific molecule into the cell. For example, to move glucose into the cell, we have to have a transport protein. And then to move fructose into the cell, which is a very similar molecule, you need to have another transport protein. Okay, so we have transport proteins that generally move one specific molecule into or out of the cell. For the outer membrane of the cell wall, we don't have transport proteins in it to move the molecules into and out of this outer membrane. What we have is a porin protein that acts as a channel. And if the cell wants the molecule to move into the cell, it will go through the porin protein. So amino acids, glucose, fructose, ATP, anything that the cell wants that's outside the cell, it'll come through the porin protein. And then the molecule has to find a transport protein to move it through the cell membrane. So the cell membrane is being very restrictive. Each protein is only moving essentially one protein, one type of, not protein, one type of a molecule. The porin protein is being less restrictive, and it's bringing a whole class of molecules through the outer membrane, many more than one. And I don't know if there are different porin proteins that do different things, or if there's only one, and it brings in all the molecules the cell wants, and of course allows molecules to leave the outer membrane if the cell doesn't want it. Any question about any of that? You'll note that there are different regions of the gram-negative cell. I'm not seeing it now. Oh, there it is, the periplasm. I don't usually talk about the periplasm. That's just the region of the cell wall where you find the peptidoglycan. There's old polysaccharides, which can be an antigen on the gram-negative cell. And we do have lipoproteins, which can be in the cell wall. And the lipid portion will be either in the outer membrane or in the cell membrane, or you may have a lipoprotein that has both, where part of the lipid is in the outer membrane of the cell wall and part of the another part of the lipoprotein is in the cell membrane. gram-negative cells, we find lipopolysaccharides. That includes lipid A as the lipid portion of that. We have lipoproteins, and we have phospholipids. The gram-negative outer membrane does protect the cell from osmotic lysis, meaning this cell, the gram-negative cell, has a cell wall, and that will prevent the cell membrane from expanding any further than the cell wall. So no further water can get into the cell while that happens. The gram-negative outer membrane can also protect the bacteria cell, like from complement proteins, let's say, and antibodies. These are proteins that the host or person make to defend themselves. And the protein has to come and bind on the cell, and if it can't bind on the outer membrane, the defensive protein will not be effective on a gram-negative cell. The gram-negative outer wall also gives protection to certain antibiotics. Penicillin G, for example, cannot get through the outer membrane of a gram-negative cell. And penicillin G has to get inside the cell wall in order for it to work. And if it can't get through the outer membrane, it can't work. So penicillin G is not effective on gram-negative bacteria. And I've already mentioned this, the old polysaccharide can give rise to antigens such as E. coli O15787. The O157 is an O polysaccharide. I've already mentioned lipid A is an endotoxin, so you need to know that. That's the important part. And we've already talked about the porin proteins are channel proteins that allow many different molecules to enter or leave the outer membrane of the cell wall. So comparing the gram-positive cell walls, they have thick peptidoglycan. The gram-negative cell walls have thin peptidoglycan. The gram-positive cell wall has a rigid structure, and the gram-negative is less rigid. It still is rigid, but it's less rigid. The gram-positive cells have teichoic acid. There's no teichoic acid in gram-negative cells. In the gram-positive cell, there's no outer membrane. In the gram-negative cells, they do have an outer membrane if they have a cell wall, and we're talking only about cell walls. And inside that outer membrane, we have lipid A, which is an endotoxin. Any question about any of that?
The connection is really spotty, so I don't know if a lot of us are getting everything you're saying. Just
ask me to repeat if something isn't coming. Like I said, I don't know what's wrong with my internet connection. It's probably the internet service provider, because according to this, it's working. I'm looking at it right here, and I don't have really much in between, and it's really only a few feet away from my computer. But it is spotty at the moment. And like I said, if you need something repeated, just ask. Okay? Sorry about that. I can't do much about it. Anything you want me repeating?
I think we missed a good amount. I don't know about anyone else, but I didn't hear for like a few minutes.
Yeah. So you missed this slide is what you're telling me?
Yes.
Okay.
I missed this one and the one before.
Oh, geez. Well, I'll talk about this one. The gram-positive cell walls have thick peptidoglycan. The gram-negative cell walls have thin peptidoglycan. There's teichoic acid in the gram-positive cell walls. There's no teichoic acid in the gram-negative cell walls. There's no outer membrane in the gram-positive cell walls. There is an outer membrane in the gram-negative cell walls. And in that outer membrane of the cell wall, there is lipid A, which is an endotoxin. All right, so let's talk a little bit about the gram stain. As you know, in the gram stain, we have crystal violet iodine, which forms a complex, which forms in the cells. In the gram-positive cells, the crystal violet iodine does not leave the thick peptidoglycan layer of the gram-positive cells. In the gram-negative cells, it's a thinner peptidoglycan layer, and the crystal violet iodine washes out with the decolorizing step. The decolorizing step also has alcohol and acetone, which can dissolve the outer membrane. Let's go back to showing you the outer membrane. Excuse me, don't know why I'm doing that. It's mainly a lipid, and alcohol will dissolve the lipid, so it'll go away. And the crystal violet stays in the gram-positive cells, so they'll stay purple in color. The gram-negative cells, after decolorization, will become clear as the crystal violet washes out. We then stain the cells with safranin, and they will pick up the pink color. So the cell wall gives rise to the gram stain. This table is showing you the differences between the two cells. And really, you can ignore all the lower part of this table, but you can take a look at the gram reaction. It's retained by the gram-positive cells, at least the crystal violet is retained, and it's washed out of the gram-negative cells. The thickness of the peptidoglycan layer and the thinness of the gram-negative layer. And this is a slight mistake there. It's thin, it's not a single layer. And you're going to have to take my word for it that the author just got this table wrong, because he's looking at the picture probably and he just said one layer, but it's not a single layer. It's a thin layer of peptidoglycan. Teichoic acid is present in gram-positive cells, absent in the gram-negative cells. You don't need to know about the periplasmic space, but you should know the outer membrane absent on the gram-positive cells, present on the gram-negative cell. And then it tells about the lipopolysaccharides, essentially none in the gram-positive cells. And there's a high content of that molecule in the gram-negative cells. There are some cells which have atypical cell walls. I want to talk about the archaea first. They are not bacteria. They're in another domain, and they do not have cell walls as I've discussed. They have cell walls, or at least most of the cells, archaea, do have cell walls, although not all of them do. But the cell walls are not made of peptidoglycan. The main molecule is either pseudomurine or another disaccharide. This is a disaccharide. So the archaea do have cell walls, or at least most of them. And there are two types. There are ones that are thick and ones that are thin. But they're not made of peptidoglycan. They're made of pseudomurine, which is another disaccharide. And it's not surprising the archaea are different because they're in a different domain. There are other atypical bacteria that have atypical cell walls. The family of mycoplasmas is an entire family of species which lack cell walls. It's curious that the mycoplasmas have sterols in their plasma membrane, which give strength and support to the plasma membrane. And if you remember, animal cells lack cell walls, and we have cholesterol, which is a sterol, in our plasma membrane to give strength and support to the plasma membrane. And the mycoplasmas are very similar. The mycoplasmas are parasites, and they infect, what do you call it, isotonic regions of animals and plants, and that's why they don't need a cell wall. Okay. And then there's one other atypical cell wall, and that's the mycobacterium. Have I shown you a picture of the mycobacterium? It is a different cell wall. It is gram positive, but it has a lot of macolic acid in the cell wall. And this macolic acid is a lipid, which makes the mycobacteria acid-fast staining, meaning they'll retain the acid-fast stain. And the macolic acid also prevents water from leaving the cell, as well as water from entering the cell. And anything that dissolves in water will have a hard time getting through the macolic acid of the cell wall. So I think I mentioned that macolic acid in mycobacteria make it difficult for the gram stain to get into the cell and then stain the cell. In my experience, mycobacterium is stained by the gram stain, but it's stained very lightly, much lighter than other bacteria. And it's because of the macolic acid does not, it helps prevent the stains, which are water soluble, from getting into the cell. I used to have a picture of a mycobacterium. Let me see if I can find that. I'll have to go online because it's either in the next lesson or in the previous lesson. I don't remember. Just be a minute. Mycobacterium. Image. Not sure which one is best, so I'll go with this one. Here's the cell membrane, number 5. And number 4 would be the peptidoglycan layer. And I think it's number 2 is the mycolic acid of the cell wall. And you can see there's a lot of mycolic acid in the cell wall. So this is gram positive cell wall, but it's very different in its structure. And that is, it has different molecules, number 3 and number 2, and probably number 1 too, which are different molecules you can find in the mycobacterium cell wall, as well as the peptidoglycan. It does have peptidoglycan, but as you can see, there's more mycolic acid in the cell wall than peptidoglycan. Let me see if this one looks at it differently. It's not a very big picture, so we'll leave it. Okay, any questions about the atypical cell walls? All right, you can remove the cell walls of a prokaryotic cell, and there's different ways to do that. Penicillin is actually one way to do it, And lysozyme is an enzyme which digests peptidoglycan. And then you can get a bacteria without the cell wall. And it's just one way to study bacteria. Actually, I don't care if you learn the names. I'm not going to test you on that. So let's move on. I've already mentioned this. The gram-negative bacteria are not as susceptible as gram-positives to penicillin G. And that's because penicillin G cannot get through the outer membrane of the cell wall of a gram-negative cell. However, don't tell me that gram-negatives are not susceptible to antibiotics. You can use other antibiotics on a gram-negative cell. For example, streptomycin is a lipid-soluble antibiotic, and it can get right through the outer membrane of a gram-negative cell wall. So streptomycin is effective on gram-negatives. It's also effective on mycobacterium. That's another story. And there are other antibiotics that can also get through the outer membrane of a cell wall. In fact, one of the early penicillins, ampicillin, is an antibiotic which can be used on both gram-positives and gram-negatives. And ampicillin can get through the outer membrane of a gram-negative cell. And if you don't know, penicillin G was the first member of the family. I think penicillin V was the second member of the family. And ampicillin was something like either the third or the fourth member of the family to be made or discovered, whatever you want to use. We'll say discovered just to be, so we don't have to get into what I'm talking about when I'm saying made. So there are other antibiotics which can be used on gram-negative cells. In fact, let me see if I can remember the one. I'm drawing a blank here on that antibiotic. There are antibiotics which only work on gram-negative cells. And there are some antibiotics which only work on gram-positive cells. Don't remember that name of that antibiotic, which only works on gram-negative cells. But there are some that only work on gram-negative and others that only work on gram-positives. And then there's many that work on both gram-negatives and gram-positives, ampicillin, carboxycillin. Most of the penicillin family members that were developed late, much later than penicillin G and penicillin V, tend to work on both gram-positive and gram-negative. All right, any questions about the cell wall? If not, it's actually 6:20. I think that's the time I'm supposed to end. So let me check on that before I end. Yep, I'm going to end it here, and I will see you at 6:30 for the lab.